Thank you all for coming. I appreciate you making the effort to come. Many thanks to Curious George for setting up the Scientia sim. And I also want to thank Shiloh for all her help that she's given me today testing it out.
In case some of you are new to the sim, I think you're aware that George is an astronomer with Caltech University Institute, actually, and he supports science, anthropology lectures, and art, and the arts here at the sim.
All right, well, why don't I get started? Again, thank you all for coming. And what I mainly want you to focus on is the central slide projector that's in the middle of this auditorium. I put up a number of displays, and I'm going to be going over them as I go through my talk. One of the things I wanted to point out on your left-hand side of the screen, you see an image of Rudolf Ruderman, who started out as a teacher and then became New York State's paleontologist in the 1920s and ended up doing some amazing work. I'll be mentioning briefly about him in this talk. But he published a monograph in 1926, so we're coming up to a celebration of his 100 years of that monograph, which basically laid out a lot about the fossils of New York. Rudolph was just amazing. He was ahead of his time. If he had computer technology, he would have even done more amazing work. So I'll say more about Rudolph later. You'll also notice to the left of the screen, I couldn't resist, but we're talking about Triarthrus trilobites. So I was able to find a 3D scan image of Triarthrus from the Paleontological Research Institute. So I threw that up there. To the right of the screen, you're going to see a trilobite. And George and I talked a lot over the last week, and I appreciate his feedback on my slides. So I thought I'd throw up that slide. So if I'm using a term for these trilobites and you're not sure where it is, just cam into that prim. And I think that will help you out. All right. We'll talk about calcite later. And I also threw up a picture of my cat who just got a toy trilobite to celebrate this talk. So anyway, go a little bit about some housekeeping issues here. And the first thing I want to say is I'm going to be talking about the classification, evolution, life history of trilobites that lived during the Ordovician. This is about 450 million years ago. And I'm planning on discussing functional morphology, or how the animals actually survive in their environments. I'm a paleoecologist. This is what I did my PhD work on. And the first thing I want to say is if you have a question or a comment that you want to make, I do have the local chat open. Feel free to ask a question. I would ask you type in caps question so I make sure it grabs my eye, because otherwise I'm afraid that I'm going to miss your question. I don't have a problem at all stopping what I'm doing. There's plenty of time. I think I've got 35 slides. So there's plenty of time to answer questions. And the other thing is, if I'm going too quickly and I'm not allowing enough time for the slides to res, then just type slow down or something in local chat so I know. All right, so let's do a little bit of an introduction here about these amazing trilobites. I first started collecting these things when I was a teenager in New York. I grew up in upstate New York. And here's a little historical perspective. This trilobite called Triarthrus was first defined in 1832 by a doctor, Dr. Green. He described the first species of Triarthrus called T. beckai. And realized 1832, we didn't know much about fossils or trilobites for that mark, but he found these things, he thought they were cool, and he did the best he could to describe it. He had a little problem, though. And the problem was that he didn't find anything as nice as what's shown in the picture there. All he got was a part of the body, thorax, and the tail of Pygidium. And as a result, there was some confusion. All right, so Dr. Green defines or puts out the name Triarthrus and assigns one species called beckai to it. All right. In fast forward 6 years, James Hall defines a new species called Triarthrus eatoni, which is sort of interesting, and we'll get into the difference between those in a minute. Then, about 10 years later, Hall decides, yeah, I don't think Triarthrus is actually a true genus of trilobite, and he decides to assign all the Triarthrus species to the Silurian trilobite Calamine, which shocked a lot of people because Calamine trilites don't look anything like Triarthrus ones do, and they're totally wrong time periods. Right. So then we go to 1892. W.S. Valiant at Rutgers College finds rare trilobites near Rome, New York. And if you look at the picture, you're seeing what one of those trilobites looks like. And they're amazing. Valiant goes nuts. He said, wow, we're not only getting the hard parts of the trilobite, but we're also getting the antenna, we're getting the legs, we're getting the appendages. This is almost unheard of in trilobites. You're lucky usually if you get just rare ones. Okay. So there's a lot of excitement about that. All right, Valiant is working near Rome at a place called Beecher's Trilobite Beds. C.E. Beecher worked at Yale University from 1895 to 1904. He loved collecting these things, and he put together large collections of these trilobites for Yale University. I mean, we're talking boxes and boxes and crates. And he started to prepare them. The tricky part was, how do you get the mud and the clay off of the trilobite without losing the delicate pyrite that records these trilobites? And the problem was that Beecher dies in 1904 before he even has a chance to clean up and prepare many of these specimens, and they're left at Yale. And Yale decides, gee, we've got so many of these boxes that we don't know what to do with them. So they're either destroyed or lost, whatever. What a terrible loss to paleontology. All right. Then we fast forward again to the 1970s. My evaluator, my mentor at Cornell University, where I was an undergraduate, studied these trilobites, and he comes up with the idea of using X-rays to study their delicate soft parts. And Cisney works out the entire anatomy of these trilobites. Everything from the digestive tract, the nervous system, the musculature system. It was an amazing study on animals that basically we didn't know much about. Then he takes it another step further and says, now that we know what their anatomy was like, we can start making suggestions about relationships. He says that these trilobites are closely related to modern-day crustaceans like crabs, horseshoe crabs, lobsters, and so on, and chelicerates like spiders. And he said they're different from insects. And the reason for that is if you look carefully at this specimen, if you look at the legs, you see how they branch. They're called biramious branches. That's typical for things like crabs and lobsters, okay, but not so much like insects that have just single appendages. And Borg's saying, boy, maybe they were tasty. You're ahead of me. Because I have no doubt that these things were tasty, especially if you like crabs. I love crab cakes. I just had them last night for supper. All right, we're talking about a fossil that was literally a textbook fossil. These are things that I studied when I was at Cornell. Triarthrus are used. They're commonly in common textbooks. You can see how they show and depict the anatomy, not only the hard parts, but the soft parts. So like I said, these are famous fossils. And the other thing that's really cool about Triarthrus is that, George has a comment. Maybe we can resurrect a trilobite. That would be interesting. Forget about dinosaurs, bring trilobites back to life. My mentor, Cisney, would love that. All right, we have a question. Where did trilobites live primarily? Deep ocean, along the ocean edges? The answer is yes, Shiloh. We have evidence that trilobites could live in environments all the way from shallow water marine environments right into deep ocean basins. And One of the things I was reading just last night was the thinking was that these trilobites were swimmers. They were sort of like sharks or whatever swimming around the ocean. That's not true. We now know that they were bottom dwellers. And in particular, if you look at the gills here, they're on the appendages. So when they're on the bottom, they spread out their gills to maximize every last oxygen molecule that's present. Yeah, curious saying then the dinosaurs, what could possibly go wrong? I don't see a problem bringing back trilobites. Yeah. Okay, yeah, they're definitely bottom dwellers is what we've got. Okay, so we'll go on to the next slide. All right, I'm going to throw around a lot of terminology here, and George has encouraged me to simplify as much as possible, but there are some terms that I'm going to have to use. So let's start off with the glabella. Everybody put their hand on their forehead. That seems to be the closest equivalent of the glabella in a trilobite, although behind our forehead is our brain. Behind the glabella of a trilobite was actually the digestive tract. These things were bottom dwellers. What they did was they would go across the bottom in oxygen, or I should say organic rich sediment. When they had a hypernome, it's like a scoop. They would scoop up the sediment and it would go along the digestive tract. They would remove all the organic matter they can, and then get rid of the rest. Some other things that we're going to be talking about are the eyes. They were attached to the head by means of the palpebral lobe. Many cases, unfortunately, we don't get the eyes, but we do get the palpebral lobes preserved. Something else we're going to discuss are the facial sutures. Trilobites were like arthropods. The way they grew was basically by bolting their outer shell. And then there are these furrows, these pits, in the glabella, and they're going to become important in a minute. All right. So those are the basic terms that you need to be aware of. Again, I resed a prim that's got all those features. So if you get a little bit lost in some of the terms that I'm using, just refer to that. That prim will be up here for the entire part of my talk. All right, I mentioned Rudolf Ruderman, and I can't say enough about this guy. Basically, Rudolf really tried to nail down the classification of these trilobites. You remember I told you that James Hall came up with two species, one Beckai and one Eatoni. You see Beckai on the left and Eatoni on the right. And Ruderman thought that one of the main ways you could tell the difference between them was the glabella, the central part. He said that Beckai, which is the ancestor, by the way, he's the first one, is sort of a square, whereas Eatoni has sort of a tall, thin glabella. All right. And that's what was used, to distinguish between these two trilobites. And what was really cool about Ruderman's work is that he's one of the first paleontologists to actually not only describe thoroughly, these trilobites, but also give quantitative measurements. He measured the length and width of the trilobites and expressed them as a ratio, so to speak. And that's what he was trying to show. But here was the problem. I started to collect more data. Ruderman only had like 10 specimens for each, for Beckai and Eatoni. And what I did was I actually calculated the ratio of length to width. And you notice here for Ruderman, his Beckai came out about 0.932, this sort of square, whereas Eatoni is 1.12. I measured more of these things and got 1.12 for the length to width ratios of the glabella for both species. So my conclusion is that you can't distinguish these trilobites on the basis of the shape of the glabella. And that's something that a lot of other paleontologists have recognized way before me. By the way, I was mentioning to George, I do have a manuscript that I'm trying to get published where I'm going to release this to the paleontological community. All right, so Beckai and Eatoni are established as species. And one of the things I mentioned is my evaluator, John Sisney, he wanted to synonymize. He said that basically, um, he thinks they're one and the same species. But what happened is that there were other workers, scientists that studied this, and they started to find the eyes that were preserved on them. And it turns out that if you look at the eye, in fact, there's a total of 6 species that were defined, mostly in 1994. Turns out there's something really interesting going on in the eye. In particular, Beckeye has a really small eye, and it's located near the front of the head. And this is something that Rudiman picked up on. He said it in 1926. And if you look at some of the other species like, let's see, where is it? Eotone, Spiniferous, Spinosus, and Rugensis. If you cam in on that picture, you'll see that the eye is getting bigger, and it's sliding also to the back of the head. And that's something that a lot of modern paleontologists have clued on. Also, the shape of the facial suture changes as you go through these species. So Rudiman said in Beckeye, the constriction of the glabella is around the first furrow. For Eotone, it's at the second. So we're coming up with a modern definition of some of these species that ties in very interestingly to their evolution. All right, so let's talk a little bit about geologic time. You can't do any evolutionary studies without a thorough understanding about geologic time. All right, and so here's geologic time scale from Harvard University, and you can see, Shiloh and I were talking about this earlier. Trilobites are very old. They lived from about 500 million years ago to about 230 million years ago. They lived way before dinosaurs came on the surface of the earth. And the ones that I'm talking about today, the Triarthrus ones, they lived around 450 million years. We're talking about animals that lived from about a 7 million year time span. By the way, Emmerloch brings up an interesting point. I'm fascinated how paleontologists can tell the difference between a new species and just a different phenotype. Really good point, and I'm going to get into that somewhat. That's been a problem that paleontologists have dealt with, and it all comes down to measure variation. We all know that in living populations, you're going to see a little bit of variability in the features of an animal, right? Okay, that's to be expected. Look at humans, for example. Think about how variable we are, and yet we all belong to the same species. And so the question becomes, when do you say, gee, I've got a group of animals that exceed the normal variation within a population, and boom, they're a new species? And I'm going to get to that later in the talk. But thank you, Emmerloch. You're just a little bit ahead of me, okay, in your question. All right, so let's talk about, again, I want to stay on the topic of geologic time. This is really important. All right, how do we know how old something is? I just said these trilobites are 450 million years old. Well, the usual way that geologists do it is with radiometric dating. They carefully measure how much radioactive material is present in a sample, and then they measure the stable material that comes from the radioactive, and they measure the ratio, and they know the rate at which radioactive material changes into the stable material. So you've got a clock that's built in. You know how much radioactive material is decayed, you know the rate at which it occurs, you can get the time. It's like if you're going on a trip, say you're going on a 100-mile trip, you're traveling at 50 miles an hour. How long is it going to take you to do the trip? Well, it's just 100 divided by 50 is 2 hours. And that's greatly simplifying what a geochronologist does, but that's the basic idea. All right, so it would be nice to be able to do radiometric dating on every rock you find, which just doesn't happen. So instead, what we do is we pick certain fossils that we know that lived during a short period of time, but they lived over great areas. And we work on getting their geologic age. And then every time you find one of these index fossils, boom, you know how old the rocks are that they are in. And it turns out that in New York State, and Rudiman picked this up early on, graptolites are a great index fossil. They're planktonic. They sort of look like jellyfish, don't they? They're floating around on the surface of the ocean. they're colonial. They've got these little things called theca that hang down from them. And in those little branches, there's like hundreds of little organisms, okay, that are just floating around. So they have a very wide distribution. They live for short periods of time. They're abundant and they're easily recognized. All right. So they make for great index fossils. Rudiman recognized 9 different zones, if you will, or time periods for these index fossils. But modern paleontologists have said, well, there's actually only 3. And George has asked me not to bore you with the Latin names, so I'm not going to. Suffice it to say, if you look at the picture here, you see the 3 main graptolites that are found in late Ordovician rocks. And they're very distinctive. Okay. You notice here on the left-hand side, Climactographis spiniferus, it's got these spines at the bottom of its theca. And then the second one doesn't have the spines. And then the third one, the spines are on the side. So based on these characteristics, you can sort of tell where you are. So I'm just going to call them zone 1, 2, and 3 as we go through, so we don't get bogged down with all the Latin names. The important thing is that these graptolites are like time markers within the rocks. And when you go in upstate New York, there's all kinds of different ways of telling time. Some of these layers you can trace over miles, so you can literally go from one place to another and know where you are. Bong says, even if you can't determine the age, you can determine the chronological order by looking at anatomical differences. Oh, absolutely. That's one of the things that's nice. If you understand the evolution, you can say something, even using Triarthrus, to determine, are these the old trilobites, medium, or young ones? And I'll get to that later on. All right, for those of you that know or are interested, here's a little bit more detail about the geology. I'm going to be talking about the Utica Shale for sure, the Whetstone Gulf. What geologists do is they put names on the rock units that the fossils are found in, and then they tie them into the index fossils, so you know roughly what the geologic age is. This is a great oversimplification of the geology, but I'm sort of blip through this because this is not geology talk. I really want to focus on the evolution of these trilobites. All right, let's understand a little bit about the plate tectonics, which goes to a question that Shiloh brought up. This is sort of a schematic diagram showing how the plate tectonics changed during the late Ordovician. So basically, you start with a volcanic island arc off the coast of North America. Think about Japan off the coast of Asia. Visualize that in your mind, and you've got a good geographic model for what it was like 450 million years ago. Well, what happens is that the volcanic island arc gets slammed across or against to Laurentia, and it forms a deep trough called the Taconic Basin. And that's where these organisms are living. It's deep, it's dark, it's low oxygen conditions. And then sediments start coming off of the volcanic island arc. You actually form a little delta. It starts filling in this deep ocean basin until finally it shallows out and it fills it entirely in. And that marks the end of the reign of Triarthrus. Once this basin gets filled in, they have nowhere to go. These are marine organisms. They can't go on land. So they go extinct after 7 million years. All right, so this shows another picture of the shallowing of this basin. The older Triarthrus tbecki probably hid from predators in the deep ocean basins. All right, and Shallow says, sorry, we can't get a digestive profile. I'm not sure what you mean. Are you talking about what these animals ate? It probably wasn't anything spectacular. I'm thinking maybe it was mostly planktonic organisms that came down from the surface. It's not like a coprolite where you can get for a dinosaur and figure out what he's eating. And I doubt there'd be much in it if you could get the digestive tract. All right, so think about this. Basically, the Triarthrus, it's got certain advantages and disadvantages living in deep water. The main advantage in deep water is that it's dark. They can hide away from predators. They have eyes, but they don't really need big ones. They have lots of organic matter in the rich sediment for food. They have some oxygen. Like I said, they spread out their appendages so they get the oxygen from the water. All right. A couple of geologists have come up with a phylogenetic tree showing the relationship of these trilobites. So we start with T. becki, which has small eyes, and then it produces T. eatoni, slightly larger eyes. And towards the end of the reign of these dinosaurs, Rogensis appears. And then eatoni splits off into 3 other species. And one of the things that I've been looking at is the mode of speciation here, and it looks like punctuated equilibrium. The evidence that I've gotten, that I've seen on these trilobites, is that once a species originates, like becki, it pretty much stays the same until it's ready to branch off, and then you get this massive, quick change in the shape of the animals to create a new species like eatoni or rogensis. It's not like slow Darwinian phyletic gradualism where there's a slow change. All right, this slide shows the location of spots, localities where trilobite data was collected. I've tried to do field work. I've found some Triarthrus fossils around Utica, New York, but for the most part, I get data from colleagues. So I've gotten a lot of information from Chris McRoberts at the State University, Cortland. Actually, I'm going to get to meet him a week from Wednesday. I've worked with the American Museum of Natural History. They've given me quite a few fossil images to photograph. I've also been to the Paleontological Research Institute, and they've given me some information. So basically, I'm working in the central part of New York, okay? We've also gotten some data from Canada on some of the species that live towards the end of the reign of Triarthrus. And this shows a data table. Basically, I put this on here just to give you an idea of the relative abundance of these species. So we get a lot of becki from some of the older rocks. We get quite a few of eatoni from the medium and later parts of the late Ordovician. And then some of the other species are just hit or miss. You know, you get a couple of specimens here or there, not a lot. I've got about a total of 100 different specimens that I've been analyzing for the research that I'm doing. I could get a lot more. Triarthrus is an extremely abundant trilobite. My mentor collected over 16,000 of these things. I know at Cortland they have 400. I tend to be a little pickier. I don't like working with material that's broken or poorly preserved, so I really culled the images down to about 100. I'd like to have more higher quality data rather than just a large number of them. All right, so let's talk about evolutions, and this will be, I know, old hat for most of you. Basically, there's phyletic gradualism. Darwin said that animals change slowly over time. Eldridge and Gould came up with a different model called punctuated equilibrium. And we also know from Darwin and so on that evolution generally proceeds by variation and natural selection. So there's this range of shapes in populations, normal variation. But what natural selection does is it says that, of course, that some of these variants are more successful in their environment, and as a result, they tend to persist. Well, it turns out there's a technique called landmark analysis that was described in Chicago by Mark Webster. I've been studying that, and it turns out it's a fantastic way for measuring variation in natural selection. And Illing has a question, were trilobites already declining before the extinction? And the answer is yes. Trilobites had their heyday in the Cambrian about 520 million years ago. They were very abundant in the Cambrian and the Ordovician, and then they start to decline. They pick up a little bit in the Devonian about 345 million years ago, and then after that, the number of genera and species tends to die off. Was there competition with other marine organisms? Possibly. Who knows? These are marine organisms. My feeling is that most likely, as you get to the Permian, about 230 million years ago, sea levels are dropping. Continental shelves where they're living on are being drained. And basically these things just died off. They couldn't move on land. For example, snails moved on land. I'm trying to think of there was some other arthropods scorpions came on land to avoid total annihilation. And she's saying, what about extreme environmental changes, low oxygen, warming oceans? Yeah, most likely they've contributed as well. Although, like I said, low oxygen may not have been so important because Triarthrus was really good at surviving under very low oxygen conditions. All right, so let's talk about methodology now, which is something that I'm interested in. What is landmark analysis? And the idea is actually pretty simple. What you do is you take an XY coordinate system, you put it on the image of a fossil like this, all right? And basically what you do is you record the X and Y position for 8 landmarks. You pick points on the shell that you think may be important. And the nice part about this is, unlike Rudiment, who only looked at the length and width of the glabella, you can look at a wide range of different parts of the head. You can look at, the anterior, the frontmost part of the head. You can put front part of the eye, the rear part of the eye. You can put the lateral side, where the occipital lobe is, where the furrows are. You can look at lots of different characteristics here of the trilobites simultaneously. And landmark analysis will show you which things are changing the fastest. The other thing that's nice about landmark analysis is you can average a large number of specimens. You can also eliminate size from the analysis. You don't want to compare a baby with an adult, so you can factor that out as a result. And by the way, this is how the landmark data looks. It's a simple text file. So it'll record the number of landmarks, the landmarks X, Y. You also have to give it a scale factor. The computer needs to know how many pixels are in a millimeter or a centimeter so that it can adjust for scale. So that's what the data looks like. It's very simple. In fact, what's really nice is if you make a mistake in the landmark analysis, for example, you're clicking on the mouse too quickly and you had, I put 9 landmarks and I only wanted 8. You can go in the text file and just delete the coordinates of the one that you didn't want and move on. All right, so let's take a look. Here's some results, and you may need to cam in on this graph to see it a little better. But you see a bunch of dots, and I've numbered the landmarks from 1 to 8. So this shows the range of variation of each feature within the trilobites that were measured. You see that around most of the landmarks, there's a circular pattern, sort of a bullseye pattern to the location of those landmarks. But if you look at landmark number 3, it's very interesting. The landmarks are skewed out, for the different specimens that we're looking. Something is going on there. They're all not clustering around one point. They're starting to be skewed towards the rear of the head. All right, so already landmark analysis is saying something interesting is going on with the eye, and in particular, the posterior, the rear side of the eye is changing over time. And one thing that you can do is you can remove all the dots. If you want to remove all the variation, you can. You can also plot up data for 2 different species. So what I did, again, if you look closely, the blue dots may be hard to see are Beckeye averages, and the red dots are the descendant, the Eotone. And if you can't see the dots, look at the arrows. 2 and 3 represent the eyes. So the eyes are moving. First of all, they're getting a little bit larger. And they're also moving to the rear of the head. So landmark analysis is cluing me into the action. Pretty much is all about the eyes in this analysis. When I was at the American Museum, they recommended that I do what's called a deformation grid on the landmarks. This is another way of looking at the data. If you look at the grid lines, they're pretty much either up or down or straight across, except for landmark number 3. There's a little bit of deformation of those grid lines. They're being bent. again, that's some indication that something interesting is going on with the rear of the eyes. Another technique, okay, that paleontologists are using is something called principal components analysis, which basically takes all the data. And this is something that George has talked about in some of his talks. It's not the quantity of data we have. We've got a lot of data, whether it's astronomy or whether it's paleontology. We have lots of information on these trilobites, and there's lots of things that we can measure about it. And what's nice about principal components analysis is it's a way of reducing the complexity of the data. We can take maybe 8 or 9 dimensions of data and crunch it down to maybe just 1 or 2. And here's what a PCA analysis looks like. And again, I know it's hard to read. And what I want to say about this is basically what it's doing is spreading out the species. All right, we got a question. Trilobites are around longer than 250 million years. Yeah. Homo sapiens, yeah, maybe 4%. Lucky to get 1%. It's very true. Trilobites were very successful in their environment. They had a hard shell to resist predators. They had a very simple lifestyle. They basically got their food from the sediment. It's pretty an easy way to go. And yeah, they hit on the success. Humans, that's going to be more on us. All right. Anyway, what I was saying about the PCA is this analysis has basically pulled the species apart by different features of the animal, in this case, the eye. So the older species are on the right-hand side of the first axis, and the younger species are on the left. This way to look at it. And then there's other differentiation that's going on here. So, and if this is confusing you, don't worry. I'm going to simplify it again a little bit further. I'm just throwing out some different ways of measuring it. This shows some of the variation. We've talked about variation before. The first 2 axes of the PCA pick up about 50%. So about 50% of the variation in this analysis is probably a result of changes in the eye. And then the remainder of that is just maybe noise in the data that's not so interesting. All right, so again, enough statistics, enough of this fancy computer stuff. What does it all mean? It's just a question that I like to ask. So basically, it's all about the eyes. So what I did was I decided that's where I'm going to focus my research, and I decided to take 3 basic measurements. And the measurements are the size of the eyes, measured from the front to the back, the field of view. Okay, and I want to say a little bit about that. Basically, it's amazing the compound eyes that have been found in these trilobites. Detailed analysis of the eyes have realized that trilobites, 500 million years ago, were able to extract calcium and carbonate from the surrounding ocean water. And they created little calcite crystals, and they arranged them so that they were perpendicular to the surface of the eye. All right, so basically they're lining these crystals up to get really good images. And over here I've got, okay, good, you can get over this part. I downloaded a calcite crystal here from PRI and also a picture. Basically, if you're not careful, if you just look through the calcite crystal, you get like a double image. But if you rotate the crystal around, those 2 images will come together. And Triarthrus was able to set up these calcite crystals so they had really good vision in the oceans. Baragon is saying the universe was able to produce trilobites. Everything since then is just evolution to detritus. Okay. Yeah, I mean, if you want to get into this, George can talk a little bit more about elements and where they're coming from that basically produced everything on the Earth. I'll leave it up to him to get into that. Okay, so what I'm doing is I'm taking measurements of the size of the eyes, field of view, and also something called the azimuth angle, the orientation of the angles relative to a vertical plane through the head. All right, and I got some very interesting results from that. All right, first off, the field of view of these trilobites increased from 56 degrees to 115 degrees. In fact, I've got some specimens where they got up to 130 or 180 degrees of field of view. That's impressive. I've talked with ophthalmologists, and they said in humans, our field of view maxes out at 90 degrees. And I can tell you as a teacher, I wish I had a field of view of 180 degrees sometimes when I was teaching, because I never knew what the kids were doing behind me. All right, so these trilobites figured it out. So one, they're increasing their field of view through geologic time. The other thing I did was I measured the size of the eyes, and Melanie at the American Museum said, be careful. It turns out that the eyes are related to how old the trilobite is. Was it a baby or was it an adult? So I correlated the size of the eyes with the size of the head. Yeah, and Shiloh and Barragan are talking about the evolution of eyes. Certainly, they appeared a number of times. I suspect that it's um, convergent evolution. You know, you've got eyes in trilobites and insects and so on, and other organisms. It's useful to see and avoid predators or to catch prey. And there's a whole thing Barragan is bringing up about light sensitivity. When I was at the American Museum, I asked, you know, when was the first eyes came up? And certainly there were eyes in the Cambrian 520 million years ago in Olin Ellis. I suspect that there were eye spots even earlier than that, maybe in the late Precambrian. Vision, yeah, as Emilok says, gives an incredible advantage. All right, so anyway, what I did was I decided to divide the size of the eyes by the size of the heads. And when you do that, you get a nice trend. T. Beckai had relatively small eyes, and they got larger to later species. Makes sense. All right. The big decision, the big thing that I found out about a month ago is that these trilobites not only made their eyes larger and increased their field of vision, but they were also able to rotate them in the head. Right, and this diagram is a little complicated, so I'll break it down. The older trilobites, the T. Beckai ones, had about a 65-degree angle between where the eyes were relative to the front of the head. Eotoni, the next variant, is 70 degrees, and Regentis, the last one, is 75. So these trilobites were able to rotate their eyes 10 degrees, okay, within their head to improve their field of view. The more you rotate, the more you see the side of the head, which I think is impressive. And what's really interesting, if you cam in on this graph, you'll notice that T. Beckai had some members of that species could get up to 78 degrees. So they were able to rotate their eyes as much as later species. It just wasn't the average. So this is a great example of natural selection in the fossil record. We're seeing a range and variation of the eyes of these trilobites, and this genus figures out that it's to its advantage to rotate more of them to the side, or natural selection, better yet, will remove trilobites that don't have maximum rotation of their eyes. All right, so what's the big deal? Why increase the size of the eyes? Why increase the field of view and rotate the angle? And the answer in my mind, I think, is simple. Somebody brought it up earlier. These guys were tasty. All right, if you look at this slide from Rudiman, 1926, you can see possible bite marks on the left-hand side of the shell. And I say possible because there's other things that are going on here. I was talking to Marcus Martin. He's concerned about molting. I think this is actual predation that we're seeing here. And I say that for a couple of reasons. First off, there were a lot of predators in these waters, and most of them were shelled cephalopods. And if you've ever seen a squid go after a crab in modern oceans, they are very aggressive. They're very fast. They will dispatch their prey within seconds. Probably having a shell on the back of cephalopods in the past slowed them down a little bit. But nonetheless, I'm absolutely convinced that these guys went after them. There were over 40 different species of cephalopods described by Hall. He probably oversplit them, would be my idea. But there are lots of these cephalopod shells mixed in with the trilobite beds. Now, one of the things that I was thinking about is what's the difference between normal breakage of these trilobite shells versus predation. And again, I'll ask you to cam in on this slide here. Basically, normal breakage of a shell happens during molting. and you'll have the head break apart at the facial suture, or they'll break between the flora of the body, or the tail break off from the body. That would be expected normally, okay, as the shell disintegrates. But what I think is evidence of predation is where you see like chew marks. These are semicircular uh, destruction of the shell that goes across flora. This is not normally along the normal breaks of the shell. To me, that tells me something was chewing on it. Um, Melanie Hopkins suggested maybe look at maybe where there's been rehealing of the exoskeleton. I've talked to Marcus, see if we can find some of those specimens. But there are lots of pictures of these things in the literature, where you see these semicircular, I call them bite marks on the shell, and it makes sense. Notice where the bite marks are. They're towards the tail or along the body, outside the range of view of this trilobite. Keep in mind, cephalopods are the smartest invertebrates in the oceans today, and they probably were pretty smart in the past. So they probably knew the fastest way to get lunch was to attack the animal from an angle that it couldn't see. All right, and here's another slide. You may have noticed some of these trilobites had spines to them. Certainly would help to avoid predators. Although the spines may have served another purpose. I saw this specimen, and it looks like the spine actually was flexible. I'm not sure why it would want to allow the spine to be able to curve. Maybe it was like some like tail or something to avoid predators. I'm not sure. All right, like I said, I've looked at phyletic gradualism versus punctuated equilibrium, a whisker, like that. Um, and it's clear the punctuated equilibrium model seems to apply to these guys. When a species comes along, it tends to stay the same for 1,000,000 years, 2,000,000 years, until it goes extinct, uh, or until it gives rise to a new species. And there's this rapid shift in the shape of the trilobites. All right, so some conclusions here. Eye size definitely increased over through late Ordovician, 450 million years ago for Triarthrus. We know the field of view greatly increased. We know the animal was capable of rotating the eyes towards the rear of the head. All of this results or increased the field of view of the trilobite and may have helped them to avoid predation 450 million years ago. One of the things I'm working on is lesson plans so that this trilobite can be used in K-12 environments. And I've got that curriculum. In fact, if you clicked on the red box during my lecture, you'll get a glass of wine. You'll also get a link to my slides, and you'll also get a link to the curriculum. So if you want to take a look at that, be my guest. And my email is wschmachterberg@gmail.com if you want to talk to me after we all log off. I don't get usually a lot of emails, but like I said, I do check that every day. So if you want to keep in touch, that's absolutely fine. And that's it for my slides. So if you have, I saw some questions and comments as we were going along, but we're good on time. I'm seeing we're at 2:52 here, so we've got time for questions. Ooh, yeah. Rhea, videos. I understand that Shiloh was telling me that this talk was videotaped, which is cool. Is that true, Shiloh? Yes, great. All right, so please send me the link, how I can get it, because there are some people that couldn't be here today. When you say videos, Rayo, I'm not talking about whether you mean a videotape of the talk, or one of the things I've looked into is morphing. And I have looked at some morphing software where you could morph, say, a Beckeye to an Eotoni or a Rogensis. I've played with the software a little bit, but I haven't actually done it. But there's a number of programs that are out there that are relatively inexpensive, and basically the way it works, Rhea, is you take the landmarks and put them into the computer, and then the software creates a series of images showing the distinction. But yeah, I've already thought about that. And like I said, email me and we can talk more. I can tell you what software I'm doing. And what we're working on is basically an open source repository of these images. The American Museum of Natural History, I suspect Cortland University, they've sent me a lot of images. I've already started sorting them out by geologic age and species, and we'd like to have those available for a download so that students, teachers, researchers can get their hands on this. The software, um, the software is available for free. You just need to have a PC. And somebody's getting into AI. I haven't done much with AI. I've played around a little bit in Unity, but that's about it. Oh, yes. Shiloh brought up about chameleons and praying mantis. Absolutely. I've started looking at them, and they're amazing. They can move their eyes by a lot quickly. Over how many degrees? It's in my paper. If you want to look at that, Shiloh, I can send you a copy of it. But yeah, chameleons are amazing. Yeah, I just have a manuscript. It's in review now. I haven't submitted it to anyone.
Well, I do have a question. So, are we seeing multiple trilobites existing at the same time, and then one dies off, and then the newly evolved trilobite with the eye movement and everything is the one which, under natural selection ideas, is that the one that continues to live and thrive? And then,
Yes.
as we have continued adaptations with the eye, then the earlier versions of the eye, even if it was moving a little bit, they die off really quick, and then you have, that's like really fast evolutionary growth, isn't it? That's amazing.
Yeah, Shiloh, here's the diagram that shows it the best. You're right. Multiple species can coexist at the same time. For example, Beckeye and Eotoni are both found in the Utica Shale. All right, so yeah, they can coexist. It's not like Beckeye gives rise to Eotoni and then goes extinct right away. No, they multiply coexist. In fact, in Canada, we have two species that coexist because of allopatric speciation, where they live in different parts. Lobsters are in the same group, I would say. There's some question. People are always arguing about the relationships of arthropods. Like I said, my advisor, John Sisney, wanted to put the trilobites with crustaceans like lobsters and crabs and spiders and separate them from insects. Melanie Hopkins said, well, they're a crown group. So what she's saying is that all these groups are monophyletic. Like they all came from a common ancestor. It's just that insects split off earlier from crustaceans. Yeah, Dally, I would say, yeah, horseshoe crabs are in with they're with crustaceans. So yeah, I would say they're related to trilobites.